Hello, everyone. I'm Anupama Jha, a postdoc in the Nobel Lab at UW. And today I'll talk about our tool, chromzoo, which does bidirectional translation between Hi-C and ATAC-seq data across species. In fact, these assays, Hi-C and ATAC-seq, capture related information about chromatin interactions. On one hand, Hi-C captures pairs of DNA loci that are proximal in 3D, and it can be visualized as a 2D contact map where each row and column represent a genomic locus. ATAC-seq, on the other hand, captures open chromatin sites in the genome, and it can be visualized as a genomic track where read pileups on the y-axis correspond to the accessible loci in the genome. With chromzoo, we aim to predict Hi-C from ATAC-seq and vice versa across many species. And this task is feasible because there is evidence that AB compartments derived from the Hi-C data correlate well with ATAC-seq track. As you can see in the density plot here for human chromosome 2, x-axis is AB compartments or eigenvectors, and y-axis is ATAC-seq, and they're correlated well with the Spearman's R of 0.67. In parallel, the DNA zoo repository has Hi-C data for over 500 species and ATAC-seq for 50 of them. Both these facts make a model that can translate between Hi-C and ATAC-seq across species desirable. Therefore, we present chromzoo, which is a deep tensor factorization model that has two inputs, a species index and a genomic position. Species index is fed into a species factor layer, and the genomic position is fed into a metagenomic factor layer. Both these factors / embeddings are shared between two networks, Hi-C-net that learns Hi-C contacts and ATAC-net that learns ATAC-seq reads. And we train chromzoo across species using both Hi-C and ATAC-seq data from all of the training species and allow all embeddings as well as neural network weights to be updated during training. However, during validation, we actually freeze both Hi-C-net and ATAC-net and only update the species embeddings and genomic embeddings for a new species using only one data modality, either Hi-C or ATAC-seq. The chromzoo model is then evaluated on its prediction of the unseen or missing data modality in this new species. To do so, chromzoo actually leverages pre-trained positional epigenetic embeddings of the human genome from a previous model from our lab, Avocado, and then it propagates this information to non-human species using one of the two strategies. The first one is a synteny-based initialization, where the syntenic locations of the non-human genomes use the human embeddings from the corresponding locations in human. And in parallel, we train a CNN model to learn these embeddings from nucleotide sequence. This model then produces these embeddings for all the non-human species using their nucleotide sequence. Once we have trained the chromzoo model, we need to compare it to sensible baselines. And for this purpose, we designed two task-specific baselines. First, for Hi-C predictions, we compute the outer product of ATAC-seq tracks after some normalization and use that as a baseline. This baseline captures regulatory interactions within A and B compartments. For ataxic predictions, on the other hand, we compute PCA over the Hi-C contact map and use the first eigenvector of PCA as ataxic predictions. In fact, the A compartments of this Hi-C eigenvector correspond with the ataxic peaks, and the B compartments align well with the non-peak regions of ataxic. So using both these baselines, we evaluate ChromeZoo first in the direction of predicting Hi-C from ataxic data. And visually, here's an example of how the ChromeZoo Hi-C predictions look like. This is Chinese hamster Hi-C scaffold 4. On top is the ground truth Hi-C, and bottom is the predicted Hi-C observed over expected values. And you can see that the predictions at the bottom capture the TAD structures, which are the majority structures that exist at 100 kb resolution. More quantitatively, we compare the synteny version of ChromeZoo model on y-axis with the ataxic outer product baseline on x-axis. And here I'm plotting Spearman R, so higher values are better. And each dot in this plot is a chromosome in a species. So ChromeZoo with synteny beats this baseline for 51 out of 153 chromosomes. But this number increases to 134 for ChromeZoo with sequence, drastically increasing our predictive performance. In the other direction, again, there is good correspondence between true ataxic reads, the track on the top left, and the predicted ataxic reads, the track at the bottom left. As you can also see with an alternative visualization of the same data on the right, the density plot showing good correspondence between true ataxic reads on x-axis and predicted ataxic reads on y-axis with a Spearman correlation of 0.72. More quantitatively, when we compare the synteny version of the ChromeZoo model with the Hi-C eigenvector baseline on x-axis, we can see that ChromeZoo beats this baseline for 31 out of 153 chromosomes, but this number increases to 76 with the sequence version of ChromeZoo. So far, I've shown you that ChromeZoo can successfully translate between Hi-C and ataxic data across 6 mammalian species at multiple resolutions. And in future, we hope to use the ChromeZoo predictions, specifically the Hi-C predictions, to find compartments, TADs, and loops using predicted data in novel species, and using predicted ataxic data to find promoters, enhancers, as well as transcription factor motifs in these new species. With that, I'd like to thank my postdoc advisor, Bill Noble, as well as the members of the Noble Lab and our amazing collaborators without whom this work wouldn't be possible. Lieberman Aiden Lab at Baylor College of Medicine, as well as Kundaje and Greenleaf Labs at Stanford. Thank you for listening to this talk.